Hello, my name is Joshua Mugabi. I welcome you to Reproduction in Living Organisms, question 1 up to 5. The graph below shows the changes in the sex hormones and the thickness of the uterine wall, obtained by close observation made by using a blood sample that were withdrawn from an adult human female at a regular interval of days by scanning the uterus respectively. The investigation was done over a period of 1 month, that is 28 days, immediately after the previous menstruation period. Our question: describe the changes in the concentration of estrogen hormone. The estrogen concentration is almost constant from day 1 up to day 8. So when you look from day 1 here up to day 8 and you look at the concentration of estrogen is almost constant. It increased rapidly from day 8 to day 12 and attained the peak concentration on the 12th day. So from the 8th day to the 12th day, it increased rapidly and it attained a peak concentration at the concentration of 12. Around here, it has reached the peak. Oestrogen concentration then decreased gradually from the 12th day to the day 24th. So from the 12th day here, it decreased gradually up to the 24th, somewhere here. And then it remained constant up to day 28. So here we can see now it remained constant from 24th up to 28th. Now let us try to describe the changes in the concentration of progesterone hormone. Progesterone concentration is almost constant from day 1 up to day 12. So let's look for progestogen. Progestogen is here. From day 1 up to around the 12th day, it is almost constant. But it increases gradually from day 16 to day 17. So from day 16th to around 17th here, it increased gradually. Then the progestogen concentration increased rapidly from day 17 up to day 23rd. So from here, 17 up to around 23rd, it increased rapidly and it attained the peak at the 23rd, around the 23rd here. It attained the peak. The concentration attained the peak at the 23rd day. Progestogen concentration then decreased rapidly up to the 28th day. So now from here, the 23rd, it decreased rapidly up to the 28th day. Part B. Compare the changes in the concentrations of estrogen and the thickness of the uterine wall. We shall start with the similarities. So we want to look at estrogen and the uterine wall. What similarities can we see? From day 4 to day 6, both estrogen concentration and the uterine wall thickness were constant. So from day 4, around here, up to day 6, up to around day 6 here, the concentration of estrogen was almost constant. The same applies here. From around there of the thickness of the uterine wall, it was almost constant. And both attained peak value at a particular time. Estrogen attained a peak value here at around the 13th day. And then the uterine thickness attained a peak value at around the 24th day. Now let us look at the differences between the estrogen concentration and the thickness of the uterine wall. Look at the differences. From day 1 to day 4, estrogen concentration is almost constant. Now if you look from here, estrogen from day 1 up to day 4, its concentration is almost constant. But from day 1 to day 4, if you look at the uterine wall thickness, it decreases slowly. So from day 1 around here, you can see that the thickness decreased gradually or slowly up to day 4. Then from day 6 to day 10, estrogen concentration increased slowly. That is from day 6 around here up to around day 10. The estrogen concentration increased slowly. Whereas in the same period, day 6 up to day 10, the uterine wall thickness is constant. From here to around here, 10th day, the thickness of the uterine wall for it remained constant. Then from day 10 up to day 12, estrogen concentration increased rapidly. That is from day 10, somewhere here, up to day 12. For estrogen, the concentration increased rapidly, and it attained the peak area on day 12. So at around day 12, the estrogen had already attained a peak. Whereas the same period, day 10 up to day 12, the uterine wall thickness, it increased slowly. It increased slowly from here up to day 12. It was increasing slowly. And it attained a peak later on day 23rd. So you see that the uterine wall attained the peak around here, and that was around 23rd day. Then from day 12 to day 23rd, the estrogen concentration decreased gradually. That is from day 12 here up to day 23rd. The concentration of estrogen was decreasing gradually up to around there. And the same period, day 12 to day 23rd, the uterine wall thickness increased rapidly. Now from here, 12th day to the 23rd, the uterine wall was increasing rapidly. Then from day 23rd up to 28, estrogen concentration remains constant. That is from 23rd to the 28th, the estrogen concentration remains constant. Whereas with the same period, 23rd day up to the 28th day, the uterine wall thickness decreases rapidly. From the 23rd here to the 28th, we can see that the uterine wall thickness is decreasing very rapidly. Part C. Let's compare the changes in the thickness of the uterine wall and progesterone concentration. We shall start with the similarities. So we can see that the changes in the uterine wall thickness is directly or closely related to the changes of progesterone concentration. Because progesterone concentration increases the uterine thickness, that is musculation and vasculation of the uterus in preparation for implantation. So we are looking now at the progesterone. We can see that progesterone concentration is corresponding with the thickness of the uterine wall. As this one is increasing, also this one is increasing. So the concentration is directly following each other. We are still on the differences. Now, you can see from day 1 to day 6, as the concentration of progesterone remains constant, the uterine thickness decreases slowly, because one of the ongoing slow thing off of the endometrial wall during the menstruation. That is from day 1 to day 6. You can see that the progesterone concentration is increasing or remains constant. And during that same period, you can see that the uterine wall thickness goes on reducing from day 1 up to day 6. What is causing this one, these changes, is because at this point here, we are having the removal of the endometrial wall, that is during the menstruation period. Then from day 6 to day 10, there is a constancy of the uterine wall thickness as progesterone concentration slowly increases due to the repair of the uterine lining following the previous menstrual flow. Beside the rise of the follicle-stimulating hormone concentration triggered by the new follicular growth. So we can see from day 6 up to around day 10th, there is a slight increase, or there is constancy. There is a slight increase in the progesterone hormone. And when you look here, there is still some constancy. The lining of the uterine wall thickness is still constant. Then from day 14 to day 23rd, we can see that the progesterone level increased rapidly. That is from day 14. Come from day 14 around here up to day 23rd. Progesterone level increased rapidly. Whereas attaining a peak at the 23rd day, because of ovulation, that produced the corpus luteum, further increased the uterine wall thickness rapidly. So when there is a high production of progesterone being produced high, we are seeing also that there is a faster increase in the uterine wall thickness because of the production of the corpus luteum, and corpus luteum produces more progesterone, and this one is the one that causes the increase in the uterine wall thickness. Then from day 23rd to the day 28th day, there was a rapid reduction in progesterone concentration, and the uterine wall thickness also reduced due to the regress of the corpus luteum. So we can see that from the 23rd here going down to the 28th, there was a decrease in the concentration of progesterone hormone. And this one also resulted in a decrease in the uterine wall thickness. This one was brought about, the reduction in the progesterone was brought about by the regress of the corpus luteum. The corpus luteum, when it regresses, that means that there is reduction in the production of progesterone. And when the progesterone concentration reduces, then the uterine wall also is going to reduce. Part D. Explain what is likely to happen between day 1 and day 6. That is, this estrogen and progesterone concentration. What is likely to happen between day 1 and day 6 at this estrogen progesterone concentration. Now between day 1 and day 6, we are seeing that the concentration of estrogen remains constant. That is from day 1 up to around day 6, meaning that it remains constantly low, that of progesterone. So it is lower, or it is higher above that one of progesterone because of the regressed corpus luteum. Why the level of estrogen level is higher than the progesterone level? It's because of the regressed corpus luteum, and that means that there is low levels of both hormones, which signals to the pituitary gland to secrete follicle-stimulating hormone that initiate follicular growth. Number 2, the table below shows the difference in the percentage of saturation of blood with oxygen at varying partial pressures of oxygen between a pregnant woman and a fetus developing in her uterus. So we have here the partial pressure of oxygen, and then we have got the percentage saturation of blood with oxygen. There is that one of the mother, and then we have the one for the fetus. Part A, plot the results on a suitable graphical form. So we have a graph showing the percentage saturation of blood with oxygen, with the partial pressure of oxygen for the pregnant mother and the developing fetus in the uterus. So this is our graph. This is the graph for the fetus from the results we have got here. This is the graph for the mother. On the y-axis we have the partial pressure of oxygen, and then on the y-axis we have the percentage saturation of hemoglobin. Compare the percentage saturation of blood of the mother and that of the fetus. Let's first look at the similarities. We can see that both percentage saturation of the mother and the fetus generally display a sigmoid pattern with increasing partial pressure of oxygen. So when you look at these curves, all of them, they are showing the sigmoid. If you look at that one of the fetus, it is showing a sigmoid pattern, and also the one for the mother is also showing a sigmoid pattern. And generally increase with the increase in the partial pressure of oxygen. So when you look at both of them, all of them, the curve, they increase with the increase in the partial pressure of oxygen. They go on increasing. And they have equal maximum percentage saturation of hemoglobin with oxygen at around 10.6 millimeters Hg of partial pressure of oxygen. So both of them, they have the same amount around here. They have the same concentration or percentage saturation of hemoglobin with oxygen at around 10.6 millimeters Hg of partial pressure of oxygen. Then you find that both of them have initially slow increase in the percentage saturation at low partial pressure of oxygen. At the beginning, when you look at them, when the partial pressure of oxygen is low, they have a low increase. Both of them, for the mother and of the fetus, they have a very low increase or slow increase in the percentage saturation at low partial pressure. And they have a rapidly increasing partial pressure of oxygen for the moderate range of oxygen partial pressure. So it increases rapidly. If you look at this point here, it is increasing rapidly for the mother, and then also for the fetus, it is also increasing rapidly at the moderate range of oxygen partial pressure. Then they have slow and almost leveling off percentage saturation of hemoglobin at higher ranges of oxygen partial pressure. So when they go on increasing, you will see that the percentage saturation at higher level goes on decreasing until when it levels off. If you look at that one of the fetus, and then you look at that one of the mother, they go on leveling slowly up to the end. Now let us look at the differences. The fetal percentage saturation, the oxygen dissociation curve, lies on the left of the mother. So for the baby, you can see the curve is lying on the left side of the mother. And then the maternal percentage saturation blood, the oxygen dissociation curve, lies on the right of the fetus. So for the mother, it is lying on the right side of the fetus. The fetal percentage saturation of blood generally is higher, except at 10.6 millimeters Hg. So when you look at it with that one of the mother, you will see that the one for the fetus, the curve is above that one of the mother, apart from only one point here where they have around 10.6 millimeters Hg of partial pressure of oxygen here. That's where they are the same. But you can see that the one for the fetus is above that one of the mother. The fetal percentage saturation of blood increases gradually for 1.3 to 2.7 millimeters Hg. So it increases gradually from 1.3 here up to 2.87 around here. It increases gradually. Whereas the maternal, it increases slowly from 1.3 to 2.7. The one for the mother, it is increasing slowly from that point up to around there. That one for the fetal, it remains constant for 9.3 up to 10.6 millimeters. So around here, it remains constant for the fetus. Whereas for the mother, from around 9.3 to 10.6 millimeters Hg, it increases slowly. For the mother, it is still increasing slowly, going up, as that one of the fetus is constant. The one for the blood reaches the maximum at a lower partial pressure. So you find that for the fetus, it is going to reach its maximum. If it reaches its maximum around somewhere here, it has reached its maximum around here at a lower partial pressure. So when you look at a point when it reaches the maximum, it is around here, around the 8 or 9, somewhere there. But when you look at the one for the mother reaches its maximum at around here, around the partial pressure of around 10 point something. Part B. Explain the differences, if any, that were identified in B1 above. Generally, the percentage saturation of the maternal hemoglobin with oxygen is lower than that of the fetus and is shifted to the right of the fetal curve because the mother, being exposed to the environment of higher partial pressure of oxygen, renders her hemoglobin a lower affinity for oxygen, but has a higher dissociation to readily supply the actively metabolizing tissues as to the fetus. So when you look at the mother's curve is shifted to the right, and that means that for it has a lower affinity for oxygen. It has a lower affinity for oxygen, but it has a higher dissociation for oxygen to easily supply the oxygen to the metabolizing tissue as well as to the fetus. For the fetus, on the other hand, is only exposed to the maternal oxygen, whose partial pressure is very low compared to the present mother's environment. Therefore, the fetal hemoglobin develops a higher affinity for the little oxygen exposed to it, but compromises the dissociation due to its relatively low metabolic profile. So when you look at the one for the baby, it is shifted to the left of the mother, meaning that it has got a higher affinity for oxygen. Any little oxygen that is supplied, it is going to be very fast absorbed by the fetal hemoglobin. And that one makes it now to become with low dissociation curve. It does not easily release oxygen because of its relatively low metabolic profile. Part C, account fully for the sigmoid shape of the maternal curve. The maternal curve is sigmoid because of the positive cooperative effect in which oxygenation of the first hem increases affinity for the second, third, and then the fourth in turn. The percentage saturation of the hemoglobin with oxygen between 1.3 to 2.7 pp oxygen, described by the lower left tail of the curve, rises slowly as hemoglobin loads more oxygen at increasing but still low ambient tissue partial pressure of oxygen. So when you look at the mother's maternal curve, here from around 1.3 to around 2.7, somewhere here, you can see that there is a lower tail of the curve which rises slowly as hemoglobin is loading more oxygen to increase the ambient tissue partial pressure of oxygen. But as each hemoglobin binds to oxygen molecule, these changes shape the globin in the way that facilitates the binding of the next oxygen. Then the further facilitates the third and that one of the fourth. Thus, there is a positive feedback effect in which oxygen loading accelerates the loading of more oxygen, accounting for the rapid rising mid portion of the curve between 2.7 to 9.3. So when you move from 2.7 to around 9.3, you will see that it increases rapidly. This is because there is already a heme group which is loaded with oxygen molecule. And when that one is loaded with oxygen molecule, it facilitates the other oxygen molecules to load oxygen very fast. That's why we have a rapid increase in the loading of oxygen from this curve here. Towards the upper right, a higher ambient partial pressure of oxygen, the curve plateaus as the hemoglobin approaches the approximately full saturation. That is, most binding sites of hemoglobin are occupied by oxygen and can't load anymore. So when it reaches here, this point, it tends to be coming to a point of plateau whereby the hemoglobin is approximately saturated and the binding sites of the hemoglobin are occupied with oxygen and cannot load any more oxygen. Part D, explain the physiological significance of the position of the fetal curve. The fetal oxygen dissociation curve shifts to the left of the maternal curve, implies increased affinity for oxygen, ensures that enough oxygen is obtained by diffusion via the placenta, permits fatty ATP synthesis and rapid growth and development. So when we look at the fetal curve, is on the left of the mother. The reason it is on the left of the mother so that it can easily absorb any oxygen that is available from the mother. Any small oxygen that is available is going to be absorbed by the fetal hemoglobin. The figure below shows the changes of some reproductive hormone immediately after conception. Compare the levels of lutein and the placental progesterone. Look at the similarities. Both concentration of lutein and placental progesterone increase up to the peak. So we have lutein. Lutein is this one here. It increases, it reaches the peak, and then it goes down. Then we have the progesterone. Progesterone is this one here. It comes like that. It rises, reaches the peak, and after reaching the peak, then it goes down completely, and it reduces completely down. Both concentrations of lutein and placental progesterone decrease rapidly after the peak. So if you look at them after the peak, this is lutein. After reaching the peak, it decreases rapidly. And also progesterone here, after reaching the peak, it decreases rapidly down. Both concentration of lutein and placental progesterone are equal at the 9th week. When you look at the 9th week, around here, they are equal. This is lutein. It comes and meets with placental progesterone. They are the same concentration. Then both begin to rising at relative concentration of 0. So all of them at the concentration of 0 around here, that's where they start rising. You can see that lutein will start rising from 0 going up. At the same, progesterone also starts from 0 rising upwards. Now let us look at the differences. Let's compare the differences between lutein and placental progesterone. Relative concentration of lutein, progesterone, gradually increases for the first 5 weeks. So here you will see that lutein progesterone, it rises during the first, it is gradual in the first week, 5 weeks, it is gradual. Whereas the relative concentration of placental progesterone, constant at 0. At 0 it is constant up to around 5 of the first 5 weeks, it is constant. The relative concentration of lutein progesterone attains a peak earlier after conception. So lutein progesterone reaches a peak earlier after conception, it reaches a peak very fast, whereas the placental progesterone attains a peak much later in conception. So you look at the placental, the one for the placental, it reaches a peak relatively higher, at a higher level. The relative concentration of lutein progesterone reduces gradually from the 5th peak to the 12th week. That is for lutein, from the 5th week to the 12th week, it reduces very gradually. Whereas the placental progesterone increases gradually within the same range. So from the 5th week here, for the placenta up to the 12th, it increases gradually here. Lutein placenta has a lower peak. If you look at the peak of lutein progesterone, it has a lower peak, whereas the placental progesterone has a higher peak. If you look at it, for it, it has got a higher peak. If you look at lutein progesterone, 0 for the period of 27th to 38th week. It is 0 from the period of 28th to 38th. It is 0. From here, it is 0 up to the 38th. 0. Whereas the placental progesterone gradually increases for the same period. Now placental here, for it, when you see, when the other one is 0, this one is gradually increasing. Lutein progesterone is 0 up to the birth time. It is 0 up to birth time. That is lutein progesterone. Lutein progesterone comes and becomes 0 up to the birth time. Whereas the placental progesterone, it peaks at the 38th week. So placenta here, it is going to peak at the 38th week here. Then it will decrease rapidly up to birth. Explain the variation of the level of human chorionic gonadotrophin hormone. Initially, 0 concentration to the absence of corpus luteum. So if you look at the human chorionic gonadotrophin hormone, it is 0 in the absence of the corpus luteum or the placenta. It is a 0. Okay? But rapidly increases from 0 to the 13th week to attain the peak. From around 0 here, it increases rapidly after around the 13th week, somewhere here. It increases rapidly and it attains the peak the 13th week, as the hormone is needed for maintenance or persistence of corpus luteum. So human chorionic gonadotrophin hormone is needed for the persistence of the corpus luteum and also the continuous secretion of progesterone. The secreted progesterone ensures proliferation of glands in the uterine walls, embryo nourishment, and cessation of any ovulation. So when we have this surge and increase in the placental progesterone, it is causing the proliferation of the glands in the uterine walls, and it is needed also in the embryonic nourishment and prevention of any ovulation to take place. The rapid decrease in human chorionic gonadotrophin level after the 13th week level was due to the development of the placenta. So this one increased rapidly and then it reduced rapidly because this placenta had been built or developed, which prompted the regress or regression of the corpus luteum. This one caused the flow down of the corpus luteum. HCG level remains low, almost constant, up to the 35th week. So when it comes down here, it remains low up to around the 35th week. 35th week, due to gradual regression of the corpus luteum. A little of HCG also produced by the placenta. The HCG concentration lowers to 0 due to placental expulsion. So when the placenta is expelled, you will see that also the HCG will go up to 0. That is after birth, when the placenta has been expelled. Because remember that the placenta also produces some human chronic gonadotrophin hormone. So when it is expelled during birth, that means there is cessation of more HCG production. Explain the variation of estrogen hormone. Now initially it is low. Estrogen is low initially and constant from 0 to the 18th week. So when you look at the initial, it is very low and it is constant up to the 18th week, because the production of from the ovaries and some from the corpus luteum, there is a negative feedback or inhibition of follicle stimulating hormone by rising concentration of progesterone. So when there is progesterone rising, there is going to be an inhibition of the production of follicle stimulating hormone. Gradual increase from the 18th to the 27th week, and rapid increase from the 27th week to the 37th week. So when you look at estrogen, from around the 27th, there was estrogen, there was a gradual increase, and then there was a rapid increase up to the 37th week. where it attains the highest peak due to increased placental activity that releases estrogen in plenty. Estrogen stimulates the hypertrophy of the endometrium cells, the formation of oxytocin receptors in the uterus, as well as promoting the development of the mammary gland in preparation for the birth. So when the estrogen is rising, its rising is going to stimulate the hypertrophy of the endometrial cells and also the formation of oxytocin receptors in the uterus, as well as promoting the development of the mammary glands in preparation for birth. From the 37th week to birth, estrogen concentration briefly remains constant, then falls rapidly of the placental expulsion, as well as the rise in prolactin levels, hormones that inhibit secretion of estrogen by negative feedback. So you can see that it rises up to the 37th week here. Then from there, it remains constant. It remains constant. When it remains constant, then it falls rapidly. It falls rapidly up to birth. The reason is that during the birth, there is inhibition of production of estrogen because of the expulsion of the placenta. And then it is also prevented by the negative feedback of the prolactin level, which keeps on rising. What are the effects of hormones, oxytocin, and prolactin towards the end of pregnancy? Oxytocin, this stimulates the myometrial contractions, and it stimulates the milk ejection from the glands, sinuses, and prolactin stimulates the mammary glands to secrete milk. So you can see that oxytocin always rises when there is birth taking place. So it stimulates the myometrial contractions and also ejection of the milk during the birth period from the mammary gland. Then you can see that the level of prolactin is also rising, and this one stimulates the mammary glands to secrete the milk. Question 4. The study was conducted on the hormonal changes in the menstrual cycle of a fertile reproductive age, periodic changes in the level of the gonadotrophin hormones, follicular stimulating hormones of the two steroid hormones, estrogen and progesterone, were studied over the 28 days of the menstrual cycle. The hormonal changes are shown in the figure below. Compare the variation of the level of estrogen and progesterone hormone. Let's first consider the similarities. In both the hormones, the level decreased from 0 to 2 days and from 23rd day to the 26th. So we are looking at estrogen. Estrogen is this one. From the first to the second, there was a reduction. It was decreasing. And when you look at progesterone, from still also the first to the second, it was also decreasing. So both of them were decreasing during that period. Then in both, the level of the hormones increased and reached the peak. Before we go to the peak, we also saw that from the 23rd, around the 23rd somewhere here, 23rd there, and the 23rd here, 24th, all of them were decreasing. Estrogen was decreasing, and then you can also see that progesterone was also decreasing. But they attained, both of them, they reached a peak, maximum peak. Oestrogen reached its peak here at around 13th, and the progesterone reached the peak at around the 21st here. In both levels, the hormones declined after the peak. So when you look at them, you will see that everyone after the peak was declining. Estrogen declined, and also progesterone after the peak, it also declined. And the level of the hormones is the same on the 17th day and on the 23rd day. So here on the 17th day, they have the same concentration here. On the 17th day, they have the same concentration, and then also on the 23rd, they are also the same. They have the same levels of those hormones. Now let us look at the differences. The level of estrogen hormone increases very slowly from 2 to 10. So from 2 to 10, if you are looking at estrogen, 2 to 10, it is increasing very slowly. And then it increases rapidly up to 13th day. Here from 10th to the 3rd day, 13th day, it increases rapidly. Whereas if you look at the progesterone, this one remained constant from the 2nd day up to the 13th day. Progesterone remained constant from the 2nd day up to the 13th day. It remained constant. Then the level of estrogen decreased rapidly from day 14 up to day 19. So from day 14 up to around 19, estrogen decreased very rapidly. But when you look at progesterone hormone, it increased rapidly from day 14 up to day 19. For it, from day 14, progesterone increased rapidly up to day 19. It was increasing rapidly. Then the level of estrogen, it has 2 peaks reached. It has got the first peak here to reach that peak, and then it reached the 2nd peak there. So it has 2 peaks. Whereas progesterone only reached 1 peak. Progesterone reached only 1 peak around there. It had only 1 peak. Then estrogen declined rapidly from day 26 up to 28th. So estrogen, you can see from 26th, it declined very rapidly up to the 28th day. Whereas estrogen, this one remained constant from the 26th to the 28th. Estrogen remained constant. That is, progesterone remained constant from the 26th up to the 28th. The level of estrogen hormone, the levels is higher from 0 to 17th day. That is estrogen. From 0 to the 17th day. If you look at it like this, coming like this up to the 17th day. the level of estrogen is higher. And from the 24th to the 28th day, from the 24th here to the 28th day, still its level is higher than that of progestogen. Whereas the level of progestogen is lower from 0 to 17. If you look at progestogen from 0 to 17, its concentration, its level is low. And then also from 24th up to the 28th day. From the 24th up to the 28th day, still its level is below that one of estrogen. The level is lower from 17th up to the 24th. That is for estrogen. From 17th here up to around the 24th, it is lower than that one of estrogen, progestogen. And also, progestogen is higher from 17th to 24th. Progestogen now for it is above the level of estrogen from 17th up to the 24th day. It attains a peak earlier on the 13th day, that is estrogen. It has its peak on the 13th day. That's where it retains its peak, and then progestogen attains a peak at the later day on the 21st. For progestogen attained its peak at the later date. Explain the relationship between the follicle-stimulating hormone and estrogen hormone. From 0 to the 3rd day, the levels of follicle-stimulating hormones increased. That one is from 0 to the 3rd day. The level of estrogen, follicle-stimulating hormone, increased. While low, the level of estrogen decreased gradually. From here, estrogen, its level decreased gradually. Decrease in the level of estrogen stimulates the hypothalamus to produce the gonadotrophin releasing hormone. So when there is that decrease in the estrogen level, it causes or stimulates the hypothalamus to produce a gonadotrophin releasing hormone, which will stimulate the pituitary gland to release follicle stimulating hormone. The gonadotrophin releasing hormone stimulates the secretion of follicle stimulating hormone from the anterior lobe of the pituitary gland. And the higher levels of the follicle stimulating hormone inhibits the secretion of estrogen from the ovarian follicles by negative feedback. So when we have a higher concentration of follicle stimulating hormone, we are going to have lower concentration of estrogen, because follicle stimulating hormone inhibits the production or the secretion of estrogen from the ovarian follicles. Then from 3 up to the 10th day, estrogen increases very slowly from the 3rd to the 10th day. Estrogen increases very slowly, while follicle stimulating hormone also is constant. If you look at the follicle stimulating hormone, for it remains constant as the estrogen is increasing. The level of follicle stimulating hormone remains relatively higher. remains relatively higher than that one of estrogen, because follicle-stimulating hormone stimulates the development of fewer ovarian follicles which secrete little amount of estrogen. So at this point where we have the high levels of the follicle-stimulating hormone above the estrogen, is because the follicle-stimulating hormone has stimulated very few ovarian follicles, and those few ovarian follicles are secreting very little amount of estrogen. That's why the level of FSH is above that one of estrogen. So from 10 up to the 13th day, you can see that the level of follicle-stimulating hormone decreased rapidly, while the level of estrogen increased rapidly. Here, from the 10th day up to the 13th day, the level of estrogen increased rapidly, whereas that level of follicle-stimulating hormone decreased rapidly. Now, this is because the follicle-stimulating hormones stimulated the development of very many ovarian follicles, which secreted a large amount of estrogen. So we can see that from the 10th day, there was high production of estrogen because the follicle-stimulating hormone had stimulated the development of very many ovarian follicles, which secreted a large amount of estrogen, which led to the very fast rise in the estrogen level and then the decrease in the follicle-stimulating hormone. The estrogen causes the repair and the growth of the endometrium. It also inhibits the secretion of follicle-stimulating hormone so that only one Graafian follicle develops. So when we have a lot of saturation of estrogen, estrogen is used in the growth of the endometrium. It inhibits the production of estrogen. It's going to be inhibited, and then also it is going to make sure that there is only one Graafian follicle that develops within the ovary. Then from the 13th to the 15th day, the level of follicle-stimulating hormone increased very rapidly. That is from the 13th to the 15th day. The level of follicle-stimulating hormone increased rapidly up to the peak, while the level of estrogen declined slowly. During that period, you can see as the follicle-stimulating hormone is rising very fast, you can see that the level of estrogen is declining very slowly. This is because the level of estrogen had reached its peak and it stimulated the hypothalamus to secrete more gonadotrophin releasing hormone, causing the temporary rapid increase in the secretion of the follicle stimulating hormone and the luteinizing hormone, and from the anterior lobe of the pituitary gland. So this reaching the peak by the estrogen stimulated the hypothalamus to secrete the gonadotrophin releasing hormone temporarily, which increased the secretion of follicle stimulating hormone, and then also it led to the increase in the production of luteinizing hormone from the anterior lobe of the pituitary gland, and that one we call it always the ovulatory surge. So the luteinizing hormone, which has been secreted, it stimulates ovulation and it stimulates the formation of the corpus luteum, which secretes progesterone from the 15th to the 16th day. So progesterone from the 15th around here to the 16th day here is being secreted by the Graafian follicle. And the level of the follicle stimulating hormone declines rapidly, while the level of estrogen continues to decline gradually up to the 20th day, where the level of follicle stimulating hormone now remains constant. So here you can see that the follicle stimulating hormone decreases rapidly up to around the 17th day and 18th, and then it remains constant. Whereas that one for the estrogen, it continues declining slowly and then it remains a little bit constant. The high levels of estrogen and increase in the concentration of progesterone inhibits the secretion of follicle stimulating hormone from the anterior lobe of the pituitary gland. Follicle stimulating hormone levels at this peak inhibit the secretion of estrogen from the ovaries. From the 19th day to the 24th day, the level of estrogen increases gradually. That is from the 19th to the 24th. The level of estrogen increases gradually, while the level of follicle stimulating hormone remains low and constant. This is because the corpus luteum secretes smaller amount of estrogen and higher level of progesterone, inhibiting the secretion of follicle stimulating hormone from the anterior lobe of the pituitary gland. From the 24th day to the 28th day, the level of estrogen declined rapidly, while the level of follicle stimulating hormone remained constant. The corpus luteum degenerates, therefore the secretion of estrogen from it ceases, and there continues to be no further secretion of follicle stimulating hormone from the anterior lobe of the pituitary gland, caused by the inhibitory effect of progesterone and estrogen. Explain the variation of the level of progesterone hormone throughout the menstrual cycle. From day 0, the level of progesterone is very low. If you look at progesterone, from day 0 up to around 14th day, it remained almost constant from day 2 to day 14. The level of progesterone remains constant within the lower levels of corpus luteum is not yet developed, and no further secretion of progesterone into the bloodstream. So during that time of 2 days to the 14th, there is no corpus luteum. Therefore, there is low levels of progesterone in the bloodstream. But from day 14 up to day 17, the level of progesterone increased rapidly. This is because the corpus luteum formed secreting a large amount of progesterone hormone. So from 14 here, we are seeing the level of progesterone increasing. This is because there is a high production of it from the corpus luteum, which is now well developed. And from day 17 to day 21, the level of progesterone increases gradually because the size of corpus luteum begins to reduce and the level of progesterone secreted reduces. That is from 17, it increases gradually because there is reduced size of the corpus luteum, which reduces the amount of progesterone being produced. Then from day 21 up to the 26, the level of progesterone declined rapidly because the corpus luteum is degenerating rapidly. Further secretion of progesterone is inhibited or reduced greatly. Complete degeneration of corpus luteum ceases the level of progesterone to remain constant on the 28th day. So from here, there is reduction in the production of the progesterone because the corpus luteum is degenerating. And when the corpus luteum has degenerated, there is no further secretion of progesterone, so it remains now constant from the 26th up to the 28th day. Predict the changes in the levels of the three hormones if fertilization occurs on the 17th day of the cycle. The level of progesterone will increase gradually and then rapidly above the level of estrogen. The level of estrogen will increase very gradually but slightly below the level of progesterone. So this one will continue to increase, and this one will also increase but at a lower level. The level of follicle-stimulating hormone will decrease gradually and remain constant. This one, follicle-stimulating hormone, will remain very low. Suggest the reason for your answer in the above. The diploid zygote formed will develop into a blastocyst. The cells of the outer layer of the blastocyst are called trophoblasts, which secretes human chorionic gonadotrophin hormone, which will maintain the corpus luteum for some time, secreting a small amount of estrogen, but later larger amount of progesterone. The role later played by the placenta when formed. Progesterone maintains the thickness of the endometrium, which sustains pregnancy, whereas estrogen stimulates the formation of receptor molecules in the endometrium sensitive to oxytocin. It will later cause birth. Follicle-stimulating hormone decreases to ensure no ovulation occurs during pregnancy and no development of the Graafian follicle take place during pregnancy. Explain the significance of the three types of hormones as fertility drugs. They can be provided to mature females individuals as synthetic chemicals that can stimulate ovulation. They either provide gonadotrophins such as follicle-stimulating hormone, which stimulates the development of the follicles, or they provide the chemical which inhibit the natural production of estrogen. And the lack of estrogen results in the production of follicle-stimulating hormone, which stimulates follicular development. Number 5, graph A and B deals with the concentration changes indicating the amount of hormones present in the blood throughout the cycle. State the three gene features of animal hormones. Secreted by endocrine glands known as ductless glands. They are entirely secreted into the bloodstream. Glands of the endocrine system secretes hormone in very minute quantities. Often the effect of these hormones is at different sites from the site of secretion. The action of hormone is on specific tissue or organs known as the target tissue or organs. They are known as chemical messengers. They modify, regulate the activity of the target tissues or organs. Most of the animal hormones are water soluble and derived from amino acids, peptides, or proteins. These molecules are hydrophilic in nature and large, so they cannot pass through the cell membrane. These hormones interact with the receptor molecules seen on the surface of the membrane. Hormones like steroid hormones, thyroid hormone, hydrophobic. They are hydrophobic. They readily diffuse through the cell membrane and bind to the receptor inside the cells. Using the two graphs in the figure above, explain the changes in the blood levels of the hormones with the time during the cycle. From day 2, day 3, concentration of both luteinizing hormone and follicle-stimulating hormone increased gradually. That is day 2 to day 3. Day 2 to day 3, they increased gradually, both the luteinizing and the follicle-stimulating hormone. Gonadotrophic releasing hormone from the hypothalamus stimulates the anterior lobe of the pituitary gland to secrete follicle-stimulating hormone and luteinizing hormone. Follicle-stimulating hormone stimulates the growth and development of the Graafian follicles in the ovary walls of the ovaries, or theca starts secreting oestrogen. And this concentration increases gradually between day 6 and 13. Secretes and day 13. The increased estrogen levels, they inhibit the secretion of follicle-stimulating hormone. When they rise, they are going to inhibit the secretion of follicle-stimulating hormone. Decrease in follicle-stimulating hormone concentration rapidly between day 6 and day 7. Day 6 and day 7. Because of the increase in estrogen, it causes the decrease in the follicle-stimulating hormone. Between day 7 and day 12, but has a light effect on the secretion of luteinizing hormone from the anterior pituitary gland. Thus, the concentration remains constant from day 3 to day 12. So this increase of estrogen has the effect only on the follicle-stimulating hormone. It reduces the production of follicle-stimulating hormone from the anterior lobe of the pituitary gland, but it has no effect on the luteinizing hormone. Therefore, for the luteinizing hormone, for which it remains constant. is not affected by the increase in the estrogen hormone. Peaking of estrogen on day 13 triggers a sudden surge in the production of both follicle-stimulating hormone and luteinizing hormone. Their concentration increases rapidly to the maximum between day 13 and day 14. That is, because of the estrogen has reached the maximum, it causes a surge of the production of luteinizing hormone and also follicle-stimulating hormone. The luteinizing hormone stimulates the resumption of meiosis in the primary oocytes to form the polar body and the secondary oocytes, which is released by the rupturing of the Graafian follicle, which we call ovulation. Estrogen concentration decreases rapidly between 13th and the 15th day. Rapid increase in the concentration of luteinizing hormone. Luteinizing hormone stimulates the remain of the Graafian follicle to develop into the corpus luteum, the yellow body, which secretes progesterone and continues to secrete estrogen. So when here the surge estrogen reaches the highest level, it causes the surge of production of luteinizing hormone. And this luteinizing hormone is going to cause ovulation to take place, and it will stimulate also the Graafian follicle to develop into corpus luteum, which Graafian follicle now will continue producing progesterone, at the same time also producing estrogen. Increasing the concentration of both hormones rapidly between day 15 and day 20. That is the progesterone and the estrogen being produced by the corpus luteum. Progesterone causes the uterus to become highly muscular and vesicular, and also inhibits the release of luteinizing hormone and follicle-stimulating hormone, decreasing their concentration rapidly between the 14th and the 15th day. Between the 14th and the 15th day, progesterone is going to cause a decline in the concentration of luteinizing hormone and follicle-stimulating hormone. The failure of the oocyte fertilization also causes degeneration of the corpus luteum, which results in the gradual decreased level of estrogen and progesterone. That is, from here, if the corpus luteum is degenerating, the level of estrogen is going to reduce. At the same time, the level of progesterone will reduce because there is less production of these hormones as the corpus luteum is degenerating. Basing on the effects of each of the hormones on the secretion of the other, show how negative feedback operates in the human menstrual cycle. The hypothalamic gonadotrophin releasing hormone stimulates the anterior pituitary to secrete follicle-stimulating hormone and luteinizing hormone. Follicle-stimulating hormone stimulates the secretion of estrogen in the ovary. Increased levels of estrogen inhibit follicle-stimulating hormone secretion and causes the secretion of luteinizing hormone from the anterior pituitary gland. Luteinizing hormone, for which it stimulates the ovulation and the development of corpus luteum, which secrete progesterone and also continue to secrete estrogen. Progesterone inhibits the release of luteinizing hormone and follicle-stimulating hormone, preventing the development of any further follicles. Suggest what would be the effect on the blood level of the hormones of if the ovary of the human is surgically removed. The level of estrogen drastically reduces. The level of gonadotrophin, follicle-stimulating hormone, and luteinizing hormone rise by negative feedback. Suggest what would be the effect of the blood level of the hormones if successfully fertilization occurred. Progesterone and estrogen level rise. Level of follicle-stimulating hormone and luteinizing hormone will reduce, preventing further maturation of the Graafian follicles and ovulation respectively. Explain how use of contraceptive pills may affect the blood levels of the hormones in the figure above. Adds more estrogen and progesterone, maintaining high level of hormones in the blood, inhibits the production of gonadotrophin hormones from the pituitary glands. State the factors that affect breeding cycle in animals. Of temperature, humidity, day length, life expectancy, genotypes, food availability, testosterone, concentration, and many others. These are some of the factors that can affect the breeding cycles in the animals. How is courtship significance in reproduction of organism? It attracts the males, possibly from a considerable distance, often by conspicuous noisy behaviors. It can drive away other mates competing for the female and oestrus. It ensures mating occurs between members of the same species and between sexually fit and healthy individuals. Synchronizes the activity of both partners for successful copulation. It induces cooperative levels of sexual arousal in both partners, making both equally ready and willing to copulate. Thank you very much for participating in reproduction in living organisms. Question 1 to 5. Please do like, share, and subscribe.